Okay, so we're going to be starting our second lecture, or week 2 lecture, and we will be speaking about membrane physiology this week. So some of the learning objectives include looking at the structure of the cell membrane. We'll talk about membrane potentials, action potentials, and also we'll be looking a bit into some of the features that we have within an action potential. Now, hopefully you guys are able to understand these very basic concepts that were covered in Physiology 1. The concept of diffusion, osmosis, what carrier-mediated transport is, what facilitated diffusion is, what primary active transport is, and what is secondary active transport. So see if you can match these up. You should be able to do this. And if not, then you have some reviewing to do. So when we look over here, again, we're looking at the cell membrane, and the cell membrane is a thin, flexible barrier. It surrounds every living cell that we have in our body. It's separating the insides of the cell from the outside. So again, it's a barrier between the inside and the external environment of that cell. Within the cell membrane, we find embedded proteins. Within the cell membrane, we also have, if you look over here, we have some of these proteins that end up going entirely across the plasma membrane. Because again, remember, this is a phospholipid bilayer, right? So you got one layer over here on top, and the other layer is down over here. So again, embedded completely across them, we call these transmembrane integral proteins. Sometimes we have proteins that are just on one side. They don't go all the way across, all right? They're on just the periphery, and we would call those peripheral proteins, right? So we have transmembrane proteins and peripheral proteins. Embedded within this phospholipid bilayer also, we have cholesterol molecules. And if you remember from Physiology 1, these cholesterol molecules are there to help stabilize this membrane, right? So they also have a lot of other functional features, but let's just say right for now, it's to help stabilize this membrane. So also we find glycoproteins that are located. So again, some of these glycoproteins, they are peripheral. They're an example of peripheral proteins. And these proteins, again, they play a very large role in the functioning not only of this membrane, but again, also they play an essential role for the function of this entire cell. All right, so there are several types of cell membrane proteins, including transporters, enzymes, receptors, as well as structural proteins. Transporters move molecules and ions in and out of the cell, helping to maintain the proper balance of substances inside as well as outside of the cell. Enzymes, if you remember, enzymes, they speed up, or again, the term that we use is that they catalyze chemical reactions, helping that cell carry out its metabolic functions. Without enzymes, these chemical reactions would take a super long time, okay? And with them, it takes like fractions of a second, all right? So let me put it this way. Without an enzyme, it may take sometimes many weeks, okay, or months for a chemical reaction to occur. But if that enzyme is present, that same reaction can occur in a fraction of a second, in a tenth of a second or less. So this is how important enzymes are. Receptors, they bind to specific molecules and allow for the transmission of signals into the cell. And this allows the cell to respond to changes within its environment. Structural proteins, they provide support as well as helping maintain the shape of that cell membrane, along with the help of, remember what we said in the previous slide? Also, that the cholesterol molecule also helps to maintain the stability of that cell membrane. So they're all working together. So overall, the cell membrane proteins are crucial for maintaining the integrity as well as the function of the cell membrane and for allowing the cell to communicate within its environment. So what are we looking over here? So the fluid inside and outside of our cells have different concentrations of certain substances. Potassium, protein, and phosphate concentrations are higher inside the cell, while sodium and chloride concentrations are higher outside the cell. Okay? So it's important for keeping the cell alive and healthy, and it happens because the cell membrane has a special mechanism for moving these substances in and out of the cell. Right? So again, key things to take away: potassium high inside the cell, sodium chloride higher outside the cell than inside. All right, moving on to membrane potentials. So let's talk about the cell membrane and how it controls the movement of ions in and out of the cell. First, let's talk about sodium. Sodium is a positively charged ion that's important for many cellular processes. However, the cell membrane is not very permeable to sodium. This means that it's hard for sodium to get into the cell. Now, with that said, the cell has a powerful sodium pump that's actively moving sodium out of the cell. This is what helps to keep this concentration of sodium inside the cell low. Now let's talk about potassium. Inside the cell, we have large and negatively charged protein molecules. These proteins attract positively charged potassium ions towards them, and this causes the concentration of potassium ions inside the cell to increase. Now, this happens because the cell membrane is more permeable to potassium and chlorine ions than it is to sodium ions. So this means that potassium and chlorine ions can more easily move in and out of the cell compared to sodium ions. Why is this important now? Well, maintaining the proper balance of ions in and outside the cell is essential for the cell to function properly. The movement of ions across the cell membrane helps to generate an electrical signal that allows cells to communicate with one another and carry out important processes, important functions. So that's the basic idea of cell membrane permeability and ion balance. So let's look at the sodium-potassium pump. So they've got, again, 1, 2, 3, 4 steps over here in this picture. It's very important to understand this. So what do we have? We have this special protein, this sodium-potassium pump. And what happens is 1, 2, 3 molecules of sodium come and attach themselves to this protein, as well as 1 ATP molecule. That's step 1. Step 2 is we end up having this ATP being phosphorylized. Okay, so we end up, when ATP gets phosphorylized, what ends up happening? Now we have ADP, okay, which gets liberated, and the sodium, this protein, it ends up changing shape. It changes shape, and it allows for this, in step 3, for the liberation of sodium from the receptor sites. Now sodium gets removed. Once this conformation of its shape takes place, next the potassium come and bind to their binding sites. Again, the next thing that will happen is this phosphorus will end up being liberated, and again we have another change of the protein shape, and the protein will change shape again, and the potassium this time will end up being released, and again this process will happen over again. You end up getting another ATP coming and another 3 sodium molecules or sodium atoms that will come and that will attach themselves onto them, and then again this process will repeat over and over again. Okay, going back over here. All right, so membrane potential refers to the electrical potential difference across the cell membrane. In other words, it's the difference in electrical charge between the inside and outside of the cell. Membrane potential arises from the distribution of ions across the cell membrane. As we mentioned earlier, the cell membrane is selectively permeable, meaning that it allows some ions to pass through more easily than it does others. This creates a concentration gradient for each ion. with a higher concentration ion on one side of the membrane compared to the other side. However, ions also have an electrical charge, which means that they're attracted to areas with opposite charge, right? So, for example, positively charged ions like sodium are attracted to areas with a negative charge, while negatively charged ions like chloride are attracted to areas with a positive charge. Right? So these two factors, concentration gradient and electrical attraction, they work together to create a membrane potential. When the cell is at rest, meaning that it's not actively sending or receiving signals, the membrane potential is called the resting potential. Right? And the resting potential is negative inside the cell. And that's typically about, well, depending on the type of cell, maybe minus 90 millivolts or minus 70 millivolts, okay, relative to the outside of the cell. Why is this important? Well, membrane potential plays a critical role in many cellular processes, including nerve signaling, muscle contraction, and secretions. So, for example, a neuron, okay, or a nerve cell, uses changes in membrane potential to transmit signals between one another. When a neuron receives a signal from another cell, it briefly depolarizes, meaning that the membrane potential becomes less negative, and this triggers the release of neurotransmitters that carry the signal to the next cell. So overall, membrane potential is a complex, important topic in this field, in our study of cell physiology. By understanding the principles of concentration gradients, electrical attraction, and resting potential, then we can start to fully appreciate how cells use electrical signaling to communicate with one another and to carry out their functions. So if you look over here, this is showing body fluids. Okay, so look, we take in water, right? Then the water that we have inside of our body, what do we have? 3 liters of water is found in plasma, okay, blood plasma. Then there's a small amount between the capillary membrane. That exchange is taking place constantly. Within interstitial fluid, okay, within the tissue fluid of our body, 11 liters of water is stored. Okay, 11 liters within tissue. Again, then we have movement of water between the cell membrane, right? Within inside the cells, all the cells of our body, look at this number, guys. It's 28 liters of water, okay, inside all the cells. So water content, it's huge, all right? So when you look over here, what do we see? We're taking in water, and also we're secreting water. Water gets secreted through kidneys, through our lungs when we're breathing, in feces when we defecate, when we perspire, when we're hot, when we sweat, water is being lost, and also water evaporates via skin as well. So over here, what we're looking at is the concentration of ions inside the cell and outside, so intracellular and extracellular concentration. So if you look over here, sodium is much, much, much higher outside the cell than it is inside. The opposite for potassium. Potassium is much higher, as we had learned earlier, inside the cell than it is outside. Same with magnesium. Magnesium much higher inside than outside. However, chloride, right, it's the opposite. It's much higher outside than it is inside. Phosphate, much higher inside than outside. Bicarbonate, much higher outside than inside. And proteins are much higher inside than they are outside. Again, over here, this slide is just showing you what are the normal levels, okay, for example, of phospholipids. Well, if you run a test, this is the normal range. It should be 280 within our body fluids, within plasma. So phospholipids, it should be within about 280, okay, milligrams per deciliter. Cholesterol, we're looking at about 150. Fats, about 125. Sugar, it should be about 100, maybe 110 is acceptable. Urea, about 15. Lactic acid, about 10. Uric acid, about 3. Creatinine, about 1.5. Bilirubin, 0.5. And then the rest of it, we should see trace levels of salts and other ions. Okay, moving on with action potential. An action potential is a rapid and brief reversal of the membrane potential in, for example, a neuron. Okay, so, and this is what enables the transmission of signals between neurons and ultimately leads to many of the complex functions of the nervous system. To understand action potentials, first we need to review the basics of membrane potential, right? So again, think back, we discussed this in the previous slides. Membrane potential, we said, is the difference in electrical charge across the cell membrane. In a resting neuron, and remember we said, depending on the nerve cell, it could be minus 90 or it could be minus 70 millivolts, right? So again, let's stick with this minus 70 millivolt example, right, for now. So in a resting neuron, let's say the membrane potential is negative inside the cell relative to outside the cell. And what is this range going to be? For now, we're going to say it's about minus 70 millivolts. Remember, it could be minus 90 also, depending on the type of cell that we're looking at. So when a neuron receives a signal from another cell, such as a neurotransmitter release from a nearby neuron, that membrane potential briefly depolarizes. This means that the inside of the cell becomes less negative, potentially reaching a level of around minus 50 millivolts or higher. If the level of depolarization is strong enough to reach the threshold, okay, it's another keyword which we'll look at, threshold. This is typically about minus 50 millivolts. Okay? The neuron is going to end up firing an action potential. All right? So, if the level of depolarization is strong enough to reach that minus 50 millivolt threshold, then the neuron is going to end up firing an action potential. Okay? So, the firing of this action potential is complex and it's highly coordinated. Okay? And it involves the opening and closing of different types of ion channels in the cell membrane. When the membrane potential reaches the threshold, voltage-gated sodium channels open up very quickly. This allows positively charged sodium ions to flow into the cell. And this is going to cause a further depolarization of the membrane potential, which then leads to the opening of even more voltage-gated sodium channels and a rapid and large influx of sodium ions. So at the peak of the action potential, the membrane potential reaches a positive value of about positive 30 millivolts. Okay? And this is due to the high concentration of positively charged sodium ions inside the cell. However, this influx of sodium ions is quickly balanced by the opening of voltage-gated sodium, not sodium, but the voltage-gated potassium channel. So let me repeat that. The influx of sodium ions is very quickly balanced by the opening of these voltage-gated potassium channels. And this allows the positively charged potassium ions to flow out of the cell, which ends up causing the repolarization of the membrane potential back towards its resting value. Okay? Now, after the action potential, the cell membrane briefly hyperpolarizes. Okay? So after the action potential, the cell membrane briefly hyperpolarizes, right? That means that the membrane potential becomes more negative than its resting value before returning to its resting potential of about minus 70 millivolts. right? Now, this brief period of hyperpolarization happens because of the slow closing of the voltage-gated potassium channels. So overall, action potentials, they're complex and they're highly regulated processes that allow the transmission of signals between neurons, and ultimately it underlines many of the complex functions of the nervous system. All right, so resting membrane potential, right? So resting membrane potential is the difference in electrical charge across the cell membrane of a neuron in the absence of any external stimulation. And to understand resting membrane potential, again, we need to start with the basic structure of that cell membrane. Right, so we need to go back to the basics. So remember, the cell membrane is made up of a lipid bilayer, right? That's separating the inside of a cell from the outside, right? So it's, in this case, if we're looking at a nerve cell, right? So it's separating the inside of a neuron from the extracellular environment. So this lipid bilayer is selectively permeable to certain ions, meaning that some ions can cross that membrane more easily than other ions can. At rest, the inside of the neuron is negatively charged relative to the outside. So what did we say before this was? We said that this is typically around minus 70 millivolts. We're going to use this for our example for the rest of this lecture. And why does this happen? It happens due to the differential permeability of the cell membrane to different ions. Cell membrane has a higher permeability to potassium ions than to sodium ions, meaning that more potassium ions leave the cell than enter it. Additionally, there are negatively charged proteins inside the cell that are too large to cross that membrane, and this contributes to the negative change of the inside of the cell. So the balance between the concentration gradients and electrical forces acting on these ions is what's determining the resting membrane potential of the neuron. The concentration gradient of potassium ions is what tends to drive them out of the cell, while the electrical gradient tends to keep them inside the cell due to their positive charge. The concentration gradient of sodium ion tends to drive them into the cell, while the electrical gradient tends to keep them outside the cell due to their positive charge. So as a result of these forces, the resting membrane potential is negative inside the cell relative to the outside. The exact value of that resting membrane potential can vary. Again, what did we say? It depends on the type of cell. Okay. Aside from that, there's other factors also, but we're not going to get into that. So again, what did we say? Minus 70, minus 90, and sometimes it may be even less. It can be as low as minus 40. So this is the range of this resting membrane potential. The range is from minus 40 to minus 90 millivolts. And one of the key things to take away again, the resting membrane potential is essential for the normal functioning of neurons, as this is what allows them to rapidly respond to the external stimulus and for it to carry out its functioning. All right, so I had told you before threshold, right? We looked at this term. So again, let's look into this in a little bit more detail and review what we've already discussed about it. So threshold refers to the level of depolarization that's required to trigger an action potential or a nerve impulse in a nerve cell. or it could be, again, a muscle cell or a gland for that matter. Okay, but again, let's use the example of nerve cells right now. So in order to understand threshold, first we need to go back and review the basics of this membrane potential. Again, this is extremely important. So if you don't understand that, then you're not going to understand threshold. All right. So as we discussed earlier on in the previous slides, membrane potential, we said, is the difference in electrical charge across the cell membrane. And we said that in a resting neuron, the membrane potential is negative inside the cell relative to the outside. And we said we're going to be using this example of, it's about minus 70 millivolts. So when a neuron receives a signal from another cell, such as a neurotransmitter that's released by a nearby neuron, the membrane potential briefly depolarizes. This means that the inside of the cell becomes less negative, and potentially it's going to end up reaching a level of around minus 50 millivolts or higher. So if the level of depolarization is strong enough to reach that threshold of about minus 50 millivolts, the neuron's going to end up firing an action potential. Now, remember, what did you say is an action potential? Action potential is a rapid and brief reversal of the membrane potential where the inside of the cell becomes positively charged relative to the outside. This triggers the release of neurotransmitters from the neuron, which then has a capacity to bind to receptors on nearby neurons and transmit the signal further. If the level of depolarization is not strong enough to reach that threshold, the neuron will not fire an action potential. Instead, the membrane potential will quickly return to its resting state, and the signal it's going to end up dissipating. So, why is threshold important? Well, it provides a mechanism for neurons to discriminate between a weak and strong signal. Okay? Neurons that receive a weak signal will not fire an action potential, while neurons that receive a strong signal will fire action potentials and transmit the signal further. And this is going to help ensure that signals are transmitted accurately and efficiently through the nervous system. All right, depolarization. So depolarization is the process by which the membrane potential of a neuron becomes more positive than its resting potential, typically as a result of an external stimulus. Now, to understand depolarization, you have to keep in mind the principles of the resting membrane potential. So remember what we said. The resting membrane potential of a neuron is negative inside the cell relative to the outside of the cell, which is about minus 70 millivolts. Why does this happen? This happens due to the differential permeability of the cell membrane to different ions, such as potassium and sodium. Now, when a neuron is stimulated by an external signal, such as a neurotransmitter or a sensory input, ion channels on the cell membrane open and allow ions to flow across the membrane. In the case of depolarization, ion channels that allow positively charged ions such as sodium to enter the cell end up being activated. As more positively charged ions enter the cell, the inside of the neuron becomes less negative to the outside, and the membrane potential becomes more positive. And this is the process of depolarization. And this can continue until the membrane potential reaches a threshold level, which is typically around minus 55 to minus 50 millivolts. Once the membrane potential reaches the threshold level, it triggers an action potential. And how do we define an action potential? We said that it's a rapid and large depolarization of the cell membrane that moves down the length of the neuron. Or again, another term is that instead of saying moving down, we use the term propagate. Okay? And it's the action potential that allows neurons to rapidly communicate with one another and to carry out complex functions such as memory and learning. All right, moving on to repolarization. Repolarization is the process by which the membrane potential of a neuron returns to its resting potential after an actual potential has occurred. Now, to understand repolarization, you have to start with the basics. So you need to understand the basic principles of an action potential. Again, this is imperative. So an action potential, we said, is what? We said it's a rapid and large depolarization of the cell membrane that moves down the length of the neuron. And we said that this ends up being triggered when the membrane potential reaches a threshold level. And what did we say that level was? We said that it's between minus 55 to minus 50 millivolts. So during the action potential, ion channels that allow positively charged ions, such as sodium, to enter the cells are activated. However, these channels are not open indefinitely, and after a brief period, they close. At the same time, other channels that allow positively charged ions, such as potassium, to leave the cell are activated. Now, as the potassium ions leave the cell, the inside of the neuron becomes more negative relative to the outside, and the membrane potential starts to return to its resting potential. This is a process of repolarization. However, repolarization does not simply stop at the resting potential. In fact, the membrane potential briefly becomes more negative than the resting potential before it stabilizes. Okay? And this is what we call, this point right over here, okay? This is what we call hyperpolarization. And this happens because the potassium channels that were activated during repolarization stay open for a brief period longer than is necessary. Eventually, the potassium channels close and the membrane potential returns to its resting potential of around minus 70 millivolts. And this marks the end of the action potential and the beginning of the refractory period. Okay. And during the refractory period, the neuron is unable to generate another action potential, right? Repolarization, therefore, we can say it's a critical process in the functioning of our nervous system. Okay. It allows neurons to rapidly return to their resting state after an action potential has occurred. All right, so let's go over a summary of the action potential. Right. So in this picture over here, they've got 1, 2, 3, 4 steps. So let's kind of we'll go over this. I think I'm going to change things around. I don't like how they kind of have this. They kind of leave this. Well, they don't leave a step out, but they don't number it, which I think they should be numbered. So what we're going to do is this. We're going to say number 1. We'll start off with this resting potential. All right. So remember, in the resting potential, step number 1. The neuron is at its resting potential. In this example, it's at minus 70 millivolts. And at this point, we have positively charged potassium ions inside the cell and negatively charged ions like chloride and proteins outside the cell. Right? Okay, then what's next? Next thing is threshold over here. Some stimulus caused that membrane potential to reach the threshold of around, in this example, minus 55 millivolts. Once that happens, then what happens is step number 2, depolarization. So this threshold triggers positively charged sodium ions to enter the cell, and it causes the membrane potential to rapidly depolarize and become more positive. Okay? And once this membrane potential reaches its peak at around positive 30 millivolts, right, now we're going to have repolarization taking place. Okay, so once the membrane potential reaches its peak, the sodium channels, they close, potassium channels, they open up, and this allows the positively charged potassium ions to leave the cell, and this causes the membrane potential to rapidly return to its resting potential. And this will complete the cycle of the action potential. Right, so be sure you review this a handful of times. Be sure you understand this concept. It's extremely important. It summarizes what we learned in detail and up to this point. All right, so in depolarization, we have what? We have an inflow of sodium, and we have an outflow or an efflux of potassium in repolarization. Hyperpolarization, we have leakage of excess potassium ions through the slow closing potassium channels. And in the fourth step, we're saying it's a return of the action potential to the resting membrane potential from hyperpolarization. This is happening due to the sodium potassium pumps. All right. More terminology. What's a stimulus? It's an applied event to a tissue that's going to produce an action potential. Okay, so some event when you apply it to tissue that will result in that tissue to produce an action potential. What's a sub-threshold stimulus? Sub means under, below, right? So it's some event that's too weak to produce an action potential. Okay, some event that's applied to tissue that's too weak to produce an action potential. Then there's a threshold stimulus. Okay, and the threshold stimulus, this would be the minimum amount of strength, the minimum amount of power. Right? Think of it that way. The minimum amount of energy that's needed, okay, for to produce an action potential. Right? It's a minimum amount of stimulus that's needed to trigger an action potential. What's a threshold stimulus? Supra means above, okay, extra. Think of it like that. Supra. This is a much bigger stimulus, or a stimulus that's more than the threshold. Right? A stimulus that's more than the threshold, that's above it, than the minimum level that's needed to produce an action potential. So if you look over here, again, what they're showing you over here is this. Look, here's a threshold over here. This is how much threshold you need, minus 55. And they're showing you, oh, look, some stimulus is being applied over here. These are all what? These are all referred to as sub-threshold stimuli. Right? All these are too little. So you're not going to get enough to cause a signal, an action potential. Right? To get the action potential, you need to have a stimulus. So that's why it says over here, failed initiation. So if you want this to occur, you need to apply enough stimulus to go from here to up to here. Okay, it needs to hit this point over here. This is that threshold. Right? So if you have a stimulus that comes up over here, then you end up getting what? Then you end up meeting this threshold. And then you're going to end up having depolarization that takes place. Okay? So over here, again, it's showing you the depolarizing phase. All right, so the depolarizing phase, this is the rising phase of the action potential. Right? This happens until the membrane potential goes up to its peak, which is, in this example, again, they're showing us about positive 30 millivolts. Right? So this is what? This is a depolarizing phase. Repolarizing phase, this is the falling phase of the action potential. Okay, it's a falling phase. So after this positive 30 has been reached, after the peak has been reached, then what's happening here is the membrane potential is going back to its resting potential. It's moving back down. Okay, and then we have this after hyperpolarizing phase, and after the resting membrane potential is reestablished, okay, we see this undershoot. Okay, so once we get to this resting membrane potential, now we have this undershoot over here, this area here. This is what's called this after hyperpolarizing, okay, or undershoot. And this typically can go all the way down to minus 90 millivolts. And why did we say this is happening? We said that this happens because these potassium channels, they're still open, they're slow to close. Okay? And this leads to this undershoot, or after hyperpolarizing phase. All right, so this is showing you what's happening with these channel ions during an action potential. All right, be sure you study this. So in the resting phase, what do we have is this. We have the sodium and potassium channels being closed. Okay, they're closed. And this causes a buildup of an equal amount of negative charges inside the plasma membrane, and an equal number of positive ions build up on the outside of the plasma membrane. Now in number 2, in the depolarizing phase, what's happening over here is that the sodium channels open up, right? So now we have the activation of the sodium channels. And when they open up, sodium moves in. As the sodiums move in through these channels into the neuron, we end up having a buildup of these positive ions. Okay? So, and this is going to continue to build up across the inside of the membrane. And the membrane is going to end up depolarizing, right? All right, now number 3, in the repolarizing phase, what's happening? Now you see over here, the sodium channel inactivation gates, they end up closing, and what's happening here? The potassium channels open up. Now the membrane starts to become repolarized as some of these ions, potassium ions, they start to leave the neuron, right? They're exiting now. So what's happening? You're starting to repolarize. You're going back down negative. This is happening over here, this red part. Okay? So moving on to the number 4, repolarization. As it continues, more potassium ions continue to enter. You start to go down even more, right? It continues to become negative. So more potassium ions leave the neuron. The negative charges start to build up along the inside of the surface of this membrane. More starts to build up. So if you look over here, you have what? Positive, positive, positive. Now what's happening? 1, 2 negatives, right? And if you look at this picture, 1, 2, and even more, 3 now. So it starts to become more and more negative, and this starts to become more and more negative also. Okay, so again, it starts to come down more. It starts to decrease further. It further repolarizes. So this will occur. This happens. This will continue until the potassium gates end up closing. So to recap again very quickly, remember at the resting phase, both the potassium and the sodium channels, they're closed. And then when you look at the depolarization phase, what do we have? We have the sodium channels that open up. Sodium channels open up. What happens? We have depolarization. So now it starts to go up, right? All right. In other words, we reach a threshold, sodium channels open up, depolarization takes place until we get to the peak, which is about positive 30 millivolts. At that point, what ends up happening is the sodium channels, they close, the potassium channels open up. Remember over here, going back to depolarization, sodium channels are open, potassium channels are still closed. Over here, both the sodium and potassium channels are closed. Right? So in resting phase, both sodium and potassium channels are closed. Number 2, sodium channels open up, potassium channels are closed. And number 3, now sodium channels are closed and potassium channels open. Now, this is what causes this. So this will occur after the peak, and now this causes repolarization. Now you're starting to get more negative, and this stays open, okay, until the potassium channels close up. All right, so let's look at the latent period. The latent period is a brief period of time that occurs between the application of a stimulus to a cell and the beginning of its action potential. Now, let's assume that we're looking at a nerve cell. So during the latent period, the nerve cell is receiving the stimulus and preparing for the action potential, but no visible change or response is yet occurring. Now, this period typically lasts only a few milliseconds, and without specialized equipment, it's very difficult to observe. So the latent period is important because it's reflecting the time that's needed, that's required for the action potential to move along, to propagate along the nerve cell's membrane and reach the axon terminal. Okay, it's a very important concept. This is showing us how long it takes for that action potential to move along the cell membrane of that nerve cell and reach the axon terminal. So once the action potential reaches the axon terminal, it's going to trigger the release of neurotransmitters, which then bind to receptors on the next neuron or muscle cell or gland and transmit the signal. So overall, the latent period, it's a critical part of the nerve signaling process, as it reflects the initial stage of neuronal excitation that eventually leads to communication between different parts of our body. All right, why and how does the action potential spread in the forward direction only? And why does not the action potential spread in the reverse direction? Let's see who knows the answer for this. Right? Something to think about. Why? And leave a comment. If you think you know the answer, post it in the comment section below. Right, here's another question. Does the action potential become weak when it reaches the end of the nerve fiber? Right, we got the answer over here for that one. An action potential at the end of the axon is identical to the original one, no matter how long the axon is. Right, so despite the length, doesn't make a difference. This way, the action potential can serve as long a distance that's needed without the signal becoming weak or without the signal distorting. Right. It's an important concept. Length does not matter. Okay, moving along to myelin. So in nerve fibers, the myelin sheath is a layer of insulation that surrounds the axon and it helps to speed up the transmission of electrical signals. The myelin sheath is made up of multiple layers of lipid-rich material that forms a barrier between the extracellular fluid and the nerve fiber. So as a result of this insulation, ions are not able to flow through this thick myelin sheath. Okay? However, we find these gaps, okay? And these gaps are called the nodes of Ranvier. Okay? So the gaps in the myelin sheath, we see one here, we see another one here, another one over here, and another one over here. These are called the nodes of Ranvier. And these are areas where the axon membrane is exposed to the extracellular fluid. So at these nodes of Ranvier, the ion channels are concentrated and ions can flow easily over here. So this means that the action potentials or rapid changes in the electrical potential of the cell membrane can occur only at the nodes of Ranvier. Right? So despite this, the action potential can still travel along the nerve fiber by a process called saltatory conduction. This means that, again, right over here, saltatory conduction. So what this says is that this implies, or this means that these action potentials, they're jumping from node to node rather than traveling continuously along the entire length of the nerve fiber. So saltatory conduction is a faster and more energy efficient method of conduction compared to conduction along an unmyelinated nerve fiber. This is because the action potential, again, as we said, it jumps from one node of Ranvier to the next rather than requiring the depolarization and repolarization of the entire length of the axon. So overall, the myelin sheath and the nodes of Ranvier, they play a crucial role in the rapid as well as the efficient transmission of electrical signals along the nerve fibers. So, another question. Which one is faster? conduction in myelinated or unmyelinated axons? Another question, which one is faster? Conduction in an axon with a large diameter or an axon with a small diameter? Right? If you know it, try to answer it in the comments below. All right, next move on to the all or nothing principle. The all or nothing principle is a fundamental property of action potentials in neurons. So it states that once an action potential is initiated, it will propagate down the entire length of the axon without any loss of amplitude or strength, regardless of the strength of the stimulus that triggered it. Okay? This means that if a stimulus is below a certain threshold, it's not going to generate an action potential at all. However, if the stimulus is above this threshold, it will trigger an action potential with a fixed amplitude that is going to be independent of the strength of the stimulus. Now, the reason for this is that the initiation of an action potential is based on the opening of voltage-gated ion channels in the axon membrane. So when a stimulus reaches that threshold, it causes that rapid depolarization of the membrane potential, which triggers the opening of the sodium, the voltage-gated sodium channels. Right? So once these channels open, sodium ions end up rushing into the cell and depolarizes the membrane even further. This triggers the opening of the voltage-gated potassium channels, and this allows potassium ions to leave the cell and repolarize this membrane potential back to its resting potential. Okay, so the all or nothing principle, it ensures that the action potentials are consistent in their amplitude and duration. That's going to allow for a reliable as well as an efficient transmission of information in the nervous system. So again, very simplistically, the all or none law is a principle that states that the strength of a response of a nerve cell or muscle fiber is not dependent upon the strength of the stimulus. Okay? Essentially, there will either be a full response or none at all. Okay, so what we're looking over here is this. Look, if you have sub-threshold stimulus, what happens? You get nothing, right? You're still at you don't reach the threshold. Remember, in this example over here, they're saying that the threshold is minus 55 moles. So if the stimulus is strong enough and you can't reach this threshold, then you do not get depolarization, right? So when you do reach the threshold stimulus, as shown over here, now we have depolarization take place. Okay, and it will go up to its peak at positive 30 millivolts, and then repolarization takes place. Now, what if you have a supra-threshold stimulus? So remember, this is showing time. This is showing the strength of the stimulus. So if we have a very strong stimulus, and if it's there for a long period of time, look what ends up happening. It still reaches the threshold and it causes depolarization. But what do we end up having? You have multiple contractions, or you have multiple depolarizations that take place, right? When you have a supra threshold of a long duration. Okay? So you don't get a much stronger, you don't see this going up very high, right? Just the duration is going to increase in this case because you have a longer duration of the stimulus that's applied compared to when you meet the threshold stimulus as well as duration. All right, so for refractory period, the refractory period is a brief period of time after an action potential is generated during which the neuron is unable to generate another action potential. Now there's two types of refractory periods. There's the absolute refractory period and the relative refractory period. The absolute refractory period occurs immediately after an action potential is generated and lasts for a few milliseconds. During this period, the voltage-gated sodium channels in the axon membrane are inactivated and cannot be reopened, no matter how strong the stimulus is. This ensures that the action potentials do not overlap or merge with one another, which could compromise the reliability of the nerve signal. So the other type, the relative refractory period, follows the absolute refractory period and lasts for several milliseconds. During this period, the neuron is able to generate another action potential, but only if the stimulus is stronger than normal. This is because the voltage-gated potassium channels are still open, which makes it more difficult for the neuron to depolarize and generate another action potential. So the refractory period is an important property of action potentials because it limits the rate at which the neurons can generate an action potential. And this prevents neurons from becoming overexcited and burning out and allows them to respond to changes in the environment in a more controlled and precise manner. So overall, the refractory period ensures that the action potentials are separated in time and that neural signals are transmitted accurately as well as reliably. So to kind of sum things up over here for this, remember, the refractory period is a period of time during which the cell is not capable of repeating an action potential. Okay? And we said that the absolute refractory period is the interval of time during which a second action potential cannot be started, no matter how large a stimulus is repeatedly applied. And the relative refractory period is the interval of time during which a second action potential can be initiated, but it requires a greater stimulus than was needed before, right? And this is called what? This is the relative refractory period. Okay, this would be over here during this phase. Okay, moving on to compound action potential. So compound action potential is a measure of the electrical activity that's generated by a group of neurons in response to a stimulus. And it's called compound action potential because it represents the sum of the individual action potentials that are generated by each neuron in the group. So when a stimulus is applied to a group of neurons, each neuron generates its own action potential, which produces a brief change in the voltage across the cell membrane. These individual action potentials sum together to produce a larger electrical signal, which can be measured as the compound action potential. Now, the amplitude and the shape of the compound action potential depends on several factors, and this includes the number and size of neurons in the group, the strength and duration of the stimulus, as well as the distance between the electrodes that are used to measure the signal. Now, one of the key advantages of the compound action potential is that it can be used to study the properties of large groups of neurons in vivo or even in vitro. So for example, scientists can use the compound action potential to study the conduction velocity of nerve fibers. Again, we're talking about the speed at which action potentials move along the axon. So overall, the compound action potential provides a powerful tool for looking into the electrical activity of neurons and understanding the complex workings of our nervous system. So by studying this action, the compound action potential, we're able to gain a better insight into how neurons communicate with each other and how this communication is disrupted in neurological disorders. All right, moving along with the cardiac muscle. So the heart is a special type of muscle that contracts and relaxes rhythmically to pump blood throughout the body. In order to do this, the heart relies on a unique type of action potential that's generated in cardiac muscle cells. When a cardiac muscle cell is stimulated, it undergoes a rapid depolarization phase that we see in other types of muscle cells, like skeletal muscle. However, unlike skeletal muscle cells, the membrane of a cardiac muscle cell remains depolarized for about 0.2 seconds before repolarizing. So during this plateau phase, the cell membrane is less permeable to ions, which helps to maintain the depolarized state. And this is important because it allows the heart to contract for a longer period of time than skeletal muscles, which have much shorter contractions. The prolonged plateau phase in cardiac muscle cells is due to the activation of calcium channels, which play a key role in the contraction of the heart. As the calcium ions flow into the cells during the plateau phase, they trigger the release of even more calcium ions from internal stores, leading to a stronger contraction of the heart muscle. Now, after the plateau phase, the cell membrane repolarizes and returns to its resting phase. This process prepares the cell for the next action potential and then allows for the heart to pump blood efficiently and effectively. So, to summarize things, the action potential in cardiac muscle cells allows the heart to contract for a longer period of time than compared to skeletal muscles. And this is what leads to effective pumping of blood throughout our body. All right, so let's summarize what we're looking at over here. Okay, so what this is showing is that in cardiac muscle, depolarization is due to sodium influx through fast voltage gated sodium channels. A positive feedback cycle rapidly opens many sodium channels, reversing the membrane potential, and channel inactivation ends this phase. Okay. Now the plateau phase over here is due to calcium influx through the slow calcium channels, and this keeps the cell depolarized because most of the potassium channels are closed, right? And repolarization is due to the calcium channels inactivating and the potassium channels opening, right? So when this happens, what's happening? Now we've got potassium efflux, right? We have a lot of potassium coming in, and this brings the membrane potential back to its resting voltage. Okay. All right, continuing with cardiac muscle. So depolarization opens a few fast voltage-gated sodium channels in the sarcolemma, which allows the extracellular sodium to enter. So this influx of sodium initiates a positive feedback cycle that causes the rising phase of the action potential and a reversal of the membrane potential from minus 90 millivolts to nearly positive 30 millivolts. So again, in cardiac muscle and heart muscle, the resting membrane potential is minus 90 millivolts. So the period of sodium influx is very brief because the sodium channel quickly inactivates and the sodium influx stops. So when sodium-dependent membrane depolarization occurs, the voltage change also ends up opening up channels that allow calcium to enter from the extracellular fluid. So these channels are what we called the slow calcium channels because their opening is delayed a little bit. So this calcium surge across the sarcolemma prolongs the depolarization, which produces a plateau in this in the tracing of the action potential. Now, not many voltage-gated potassium channels are open at this point, so the plateau ends up being prolonged. As long as calcium is entering, the cell continues to contract. Okay? So as long as you keep the calcium, these calcium channels open, calcium is going to keep coming in. The cell's going to contract, right? And this develops the muscle tension, okay, during that plateau. And it peaks just after the plateau ends. So after about 200 milliseconds, the slope of that action potential, when you look at the tracing, it falls very quickly. It rapidly starts to fall. That's what you see over here, right over here. Okay. This root repolarization happens from inactivation of the calcium channels and opening of the voltage-gated potassium channels. Okay, so go back and look at the previous slide in case there's still confusion, because it shows it out very nicely. So again, the rapid loss of potassium happens when the potassium channels open up, right? And then you get rapid repolarization that takes place. The cells in the rhythmical control center of the heart have a resting membrane potential that it's not negative enough to keep all the sodium and calcium channels closed. So this means that some sodium and calcium ions can flow into the cell, causing the membrane voltage to become more positive. When the membrane voltage becomes positive enough, the action potential is generated, which is necessary for the heart to beat. So this process is called the re-excitation process, and it allows the cells in the heart to generate their own electrical impulse and maintain a spontaneous rhythm. So as we said in the previous slide, the resting membrane potential isn't enough to keep the sodium and calcium channels completely closed. So because of that, some sodium and calcium ions flow inwards, and this causes the membrane voltage to increase in the positive direction. And this leads to the generation of an action potential. Now, after the action potential, the membrane repolarizes. However, after a delay of a few milliseconds or seconds, spontaneous excitability occurs, and this causes depolarization again, which results in a new action potential. So this cycle continues repeatedly, and this leads to self-induced rhythmic excitation of this excitable tissue. So in summary, the re-excitation process in the heart's rhythmic control center is needed for spontaneous rhythmicity. Okay, it's essential, it's necessary. So the influx of sodium and calcium ions leads to the generation of an action potential, and the subsequent depolarization and repolarization cycles cause self-induced rhythmic excitation of heart tissue, okay, of these cardiomyocytes. That's it for this week's recording. Thank you for viewing this.